Hi, today's topic is pharmacognosy. The term pharmacognosy was first introduced by German scientist Seidler way back in 1815, and it is actually knowledge of drugs, that is made up of two words: pharmakon, that is drug, gnosis is knowledge, and the knowledge of pharmacognosy was developed slowly as the man progressed, or the ancient civilization progressed from the primitive man. Man actually came to know by looking around its surroundings and the other animals which were already there, and he was actually learning lot many things from these animals, and from that he gained the knowledge that there are certain plants which can be used as healing medicines, or to eliminate pain, or to control suffering and counteract diseases. And thus actually the process, or the journey of pharmacognosy started. By the trial and error, you can say, of primitive man must have acquired biological knowledge about the healing, and as I said, the curing properties of the plants, and up to certain extent some animal parts or some minerals. That is how the herbo-mineral system must have developed in pharmacognosy. One such very good example is of Ayurveda. Second is of Chinese medicines, and maybe the third example is Egyptian medicines, so on and so forth. These observations of human beings were initially passed on from one generation to another generation, just by saying, or mouth to mouth, right? So one generation passed on that to the next by just telling about the qualities of certain plants, and maybe that is how the knowledge sharing must have started in olden days. At that time actually there was no extraction, separation, isolation and identification which was done by human beings, because the knowledge was in very primitive stage. But later on, as the science progressed, as the pharmacognosy progressed, extraction, separation and isolation, as well as identification, gain lot of importance, and thus actually the development of pharmacognosy must have gained momentum. This is how the scientists define pharmacognosy. You may actually call it the history, preservation and uses of drugs and their commercial uses. So that may be one kind of pharmacognosy definition. And second, maybe pharmacognosy is an objective study of crude drugs of plant, animal and mineral origin that have been treated scientifically. Then comes how the classification must have taken place, classification of drug, how they must have classified, or how the classification system must have evolved. The first and very old type of classification of drug is an alphabetical classification. By name you can classify the drug. Secondly, as the knowledge developed, or knowledge was added day by day, or year after year, man came to know about their taxonomy or taxonomic position. Depending upon that, the taxonomic classification came into existence. Scientists used to use the classification to classify drugs depending upon their families, or depending upon their genus and species, etc., etc. Morphological classification is which part of the plant or animal is useful, or how the mineral looks like in its natural form. So that is how actually the morphological classification was developed. Next important classification is by their pharmacological or therapeutical action. Whether it is purgative, whether it is antihelminthic, and so on. So they were actually classified on their pharmacological or therapeutical action. Then came the very important system of classification, that is chemical classification. Obviously, what kind of chemicals, or what type of chemicals these are, just like alkaloids, glycosides, tannins, volatile oils, etc. So this is how they were classified. And the last one, the most recent one, is chemotaxonomical classification, where chemical nature as well as its taxonomical reference is taken into consideration, and the drugs are classified on the basis of that. As we all know that drugs, especially from plant origin, we use, they are nothing but the secondary metabolites which are produced within a plant body. They are usually not essential for cell growth as the primary metabolites are, but they are involved in defense mechanism, or maybe in the process of pollination. Usually end product of the primary metabolites are derived from them, so they are not directly synthesized within a plant body, but it is a byproduct, or it is an end product of some primary process accumulated in small quantities. So if you try to extract this kind of drugs from the plant, they will be accumulated in small quantities, and its absence usually do not lead to death of plant, as it is in the case of primary metabolites. Some examples, as I've already told you, are on the screen. So when the knowledge is developed, there is always a need to document that. So that documentation started all over the world as making pharmacopoeias. So pharmacopoeias were first written by many of the advanced countries like US or UK, or maybe Russia, or maybe Australia or Japan. Indian Pharmacopoeia was actually the committee established somewhere in 1948, but the Pharmacopoeia came into existence in 1955. Till that time, Indian government used to used to follow British Pharmacopoeia for their purpose of manufacturing drugs from plants as well as from any such natural source. And see, on the screen you can see there is British Pharmacopoeia, Japanese Pharmacopoeia, there is US Pharmacopoeia as well, and Indian Pharmacopoeia are there are different types of Indian Pharmacopoeia, like Indian Pharmacopoeia and Indian Herbal Pharmacopoeia, Indian Ayurvedic Pharmacopoeia, etc., etc. So pharmacopoeia are basically used for identification of the material, determination of their quality and or checking their quality and purity, and if adulterated with some other plant, then what may be the probable adulterant. That everything is included within a pharmacopoeia. The authenticity of crude drug is established by referring to its description as given in the pharmacopoeia. It is its taxonomical description, as well as its chemical analysis, the chemical test which can be applied for the active constituents of the plant can be found in the pharmacopoeia itself. There are certain numerical standards or numerical values which are given for certain chemicals or certain parameters of the drug, and which can be very helpful for establishing purity and quality of the drug. The government of India, as I told you, constituted its pharmacopoeia committee in 1948, and first pharmacopoeia came in 1955. IDMA, that is Indian Drug Manufacturers Association, and ADMA, Ayurvedic Drug Manufacturers Association, were some of the key bodies which were involved in preparation of Indian pharmacopoeia. Indian pharmacopoeia includes methods of analysis for various drugs, then classical procedures, instrumental techniques, modern techniques such as spectroscopy and chromatography, and it includes monographs for almost all drugs from Indian origin, and in the form of monographs, which includes English name, its synonyms in different languages of India, including Sanskrit, and its chemical names, descriptions, color, taste, solubility, purity, standards, everything is given in that particular pharmacopoeia or in that particular monograph. This is how the pharmacopoeia becomes very useful document for industry to manufacture many of the drugs, and it will be a helpful document for authentication of the drugs. Thank you for the patient listening.